So I am Igarashi. I'm associate professor in the Department of Anatomy and Neurobiology, but I'm also affiliate in UCI Mind. So we do olfactory memory using rodents. And our question is how this memory of olfaction is made in healthy animals. But and then so we study circuit mechanism of the brain to make this item memory of olfaction. But at the same time, we want to use this idea, this knowledge, to understand how this circuit mechanism is degrading in Alzheimer's disease. So we do both ways. We make research on item memory. In this case, item is olfaction for a memory of I mean, or odor cues for rodents, because odors are pretty what rodents can remember very well. There are three types of memory: memory of when, memory of what, and memory of where. These three components of memory is supposed to be done in different circuits in the brain, but we are specialized in item memory, and we found that this item memory, of a memory of olfaction, is done in the brain area called the lateral entorhinal cortex. So we are focusing on the lateral entorhinal cortex from when I was a postdoc in Norway. I was a postdoc in the lab of two couple of professors called Edvald and May-Britt Moser. They got Nobel Prize in 2015, but they are focusing on where memory, so memory of place. But when I was a postdoc, I started to work on item memory using olfaction, and we are focusing. I was focusing on the lateral entorhinal cortex. So it's a really long-term process of research. We recently found that this lateral entorhinal cortex is separating the memory of olfactory memory into the memory of odors that is associated with reward, and against to the odors that are associated with punishment. So when animals are on a task where they need to lick for odor cues, so there are two types of odor. Let's say odor A and odor B. Odor A, when animals are presented with odor A, and then animal can lick for sucrose water. But when odor B is presented, animal cannot lick because otherwise he will be given with bitter water, so it's a punishment. Then animals start to separately memorize them. Then when animals are remembering this task, we found that the cells, neurons in the entorhinal cortex, is separating these two types of cues into different category. So there are two types of cells that fire to either A or B. That's what we found. Whatever we found in basic research, we can turn these findings into Alzheimer's disease. The strength of animal research is that we can make research in really in detail, so that you can, to the level that you cannot ask in, for example, of course, human, because we cannot do experiments in human. But for example, we found two types of cells. Then the question will be, are these two types of cells are just there in Alzheimer's mouse model? We found that they are disappearing. We also found in healthy animals that the mechanism to separate these two cells are actually a chemical substance called dopamine that is applied to the entorhinal cortex. So then the question is that these dopamine is existing in Alzheimer's disease. And then we checked the Alzheimer's disease mouse and then found that the dopamine is really reducing in Alzheimer's mouse model. So that is, we think it's very new. We are right now trying to publish that paper. We really want the clinician to check that check if the dopamine in the entorhinal cortex is reducing or not, because no clinical researcher really asks that question. So our role as a basic researcher is to bring out a new idea to clinicians, to say like, hey, please ask this, please investigate this. I think that's our role. We have so many talented researchers in our lab. Technicians, grad students, undergrads, postdocs. So we do in team, and they're really doing a great job.